Morning, ladies and gentlemen. My name is Kansti Garma. I'm a second-year PhD student at SMLS University, and my thesis topic is the role of microRNAs in renal fibrosis and their molecular pathway. Today, I'm going to introduce 12-month progress report until August 29. My supervisor is Gabor Kukeni, and you can also see our team here at the Central for Translational Medicine. And my vision is, in general, I would like to contribute to building healthy communities, which is mainly supported by evidence-based and translational medicine. And my doctoral study, my short-term mission is to investigate novel dynastic microRNA, which may play an important role in renal fibrosis. And based on my vision and mission, I started two specific projects with the team at a center, and one is started in September last year, one is started in October. Now, I would like to introduce the more detailed information about my two projects. The first one is systematic review and meta-analysis. The title is microRNA biomarkers in early and late stage of chronic kidney disease. And you can also see our team here. And our manuscript status for this project is we already submitted our manuscript and waiting for response from the journal, and the journal is Kidney International. And for this project, now I would like to introduce a short background why we have to study microRNA and chronic kidney disease. Actually, I would say kidney disease is a silent killer because 10% of the world population is affected by this kidney disease, and those people can lose up to 90% of kidney function without experiencing any severe symptoms. And we're willing to find and improve the management and diagnostic approach of this disease. And in the last 15 years, microRNAs were considered the potential diagnostic, also the therapeutic biomarkers. And it has a potential to target the genes and inhibit its function and also its effective protein expression. And the scheme, you can see how the microRNA interacts with other molecules and other molecular pathways. Even it can interact with the drugs and chemical substances. And so it also plays an important role in any kind of disease. And so right before we start the microRNA related meta-analysis, we will also consider this a background, and we asked the specific question, which was, it was like, which are the most distributed microRNAs in human and animal CKD? And we set the PICO framework according to our instruction, and we, at the end, we hypothesized that maybe chronic kidney disease is characterized by this distributed microRNAs from providing assays in human and animal. And in here, in this slide, we show you the methods that we used in our meta-analysis. There is a thousands of articles that were focused on microRNA kidney disease, but we were only focused on array data and also sequencing and also PCR. But PCR has to be like more than 96 well played. And at the beginning, we had 17,000 articles. And finally, we had a human 32 and animal 13 studies included in our meta-analysis. And the robust aggregation was the main statistical analytical method for us. In order to perform this analysis, we cleared several lists, microRNAs, and ranked them. And also we used R program to make an analysis. And after that, we determined the target genes of these microRNA signatures. And after that, we performed the in silico analysis to define the molecular pathways that's related with this microRNAs in human and animal. And now I'm happy to introduce the results section. Most analyses will be performed from robust aggregation, and outcome will be up or down with microRNAs, and there will be 3 different subgroups. And for human studies, we had 1 to 3 subgroups based on sample types. And for animals, we had only one group that is basically based on kidney tissue and also the different kidney disease model, for example, diabetes and UO model. And this is the typical example of our results. And it's from Robertson aggregation. It's the most upregulated microbes in mice kidney tissue. There were 6 microRNAs were upregulated. That's colored by red in this heat map. And actually, this is the typical analysis that we performed in each subgroups. And so, in order to save the time, now I'd like to introduce the summary results. And here you can see the all microRNA signatures by human and mice by different sample types. On the left side of the screen, you can see in the urine samples that were 4 microRNA were down, 1 is up. In a blood, 2 is down, 1 is up. In a kidney tissue, there were 3 is down, were down regulated. For mice, diabetic kidney tissue disease model, there were only 2 were down, UL model, renal fibrosis. In here, there were 5 up and 1 were down regulated. And since we are performing the different subgroup analysis, we are also interested in what will happen early and late stage of kidney disease. And interestingly, in early stage, there were only 2 microRNA were upregulated in urine and blood. In the late stage, several microRNAs were downregulated significantly. And based on our microRNA signature investigation, we can also determine the relationship of microRNA, gene, and disease. For example, microRNA 181A5 is located to chromosome 1, and the deletion of this gene may also have a correlation with immunoglobulin A and nephropathy. And so we can also investigate this information more in detail based on our analysis. And this bar chart will show you the different sample types and also microRNAs and the number of target genes. We carried out these target genes to determine the molecular pathways. This is the example of pathway enrichment analysis. It's from animal kidney tissue, and there were fatty acid prime ECM receptor interaction, and the steroid biosynthesis pathways were most enriched in animal kidney tissue model. And in conclusion, we specified sample, biological sample specifically, and the stages of microRNA signature in chronic kidney disease, and also we checked the translation of marine model of kidney disease to the human side, and also we investigated molecular pathways that have a potential influence on the kidney disease, and that's derived from microRNA signature data. And also we had study implication in clinical and translation side. And clinical side, we are hoping to bring up the candidate microRNAs in further clinical investigation, and also we specify the sample specific microRNA signatures, for example, in blood and urine, and also we had translational implication too. And we had also strand enemy. Basically, we were working with the providing studies. There were lack of raw data, and we will only consider the mouse data for animal studies. And here I would like to introduce my second project, which started on October 2021. It's also systematic meta-analysis. You can also see our team here. And the title is Diagnostic Accuracy of MicroRNAs in Chronic Kidney Disease. And we were planning to submit in November this year. Basically, it's the pure clinical diagnostic study, and we asked the specific question: what is the diagnostic accuracy of microRNAs in humans diagnosed with chronic kidney disease compared with healthy individuals? And we set the PICO framework. PICO was healthy and sick human tested for microRNA, and exposure and comparator was the microRNA expression. Outcomes was full sensitivity, specificity, and ROC and AUC curve. We also have a clinical implication for that. We will know the diagnostic of microRNAs in kidney disease, and we'll be the second team that's determined as the accuracy in kidney disease in the world. And we did systematic search accordingly. And this slide will show you the progress of this project. We finished the data extraction. Now we are planning the statistical analysis. And at the end, for the project overview, one project is submitted and one is on the statistical analysis part, and we're planning to submit on November. And thank you very much for your attention. And I would like to close up my talk by saying Mongolian proverb. It's in English translation means, where there is a will, there is always a way. Thank you.